still 6 students is not in our class, but we will not wait for them. So we will start our class. Today we will in our medical genetics, we learn about the human disease of the single gene disorder, polygenetic disorder, population disease, and mitochondrial disease. Then the last genetic disease is the chromosome disease. But before we learn about the chromosome disease, we will learn some knowledge of human chromosome. Maybe you have learned some knowledge of the chromosome in your cell biology or in molecular biology, biology, chemistry, and molecular biology. But in our medical genetics, we will learn some other knowledge of the human chromosome. Maybe some knowledge you have learned, but some knowledge for the chromosome for in chromosome disease abbreviation, maybe you have not learned. So first, what is chromosome? Here from picture, we can see this is a chromosome and with 2 chromatids. But the gene is a functional DNA sequence and his flanking sequence. And the gene is located on the chromosome, but the chromosome have proteins except DNA sequence. So the chromosome is a carrier of genetic material, the genes or the DNA sequence. But the chromosome is made up of DNA and proteins and has the function of storing and transmitting genetic information. So the chromosome contains DNA sequence and proteins, and most of the proteins is a histone. Here, this picture saw a nucleus and a microscopy. It is a picture in my experiment, and the nucleus is dyed with gem cell. And you can see in this picture, in the nucleus is many dark staining area. It is called the chromosome. And the chromosome is an important marker for species identification. For example, because different species have different chromosome number. For example, the human has 46 chromosomes, and the fruit fly has 8, and the rat have 42, and the fin duck has 80, and the tobacco has 48. The belly has 14, and the watermelon is 22, and the bean is also 22. So, according to the marble in a species, maybe you can have some knowledge what species they are belong to. But here you should know we human beings have 46 chromosomes and 23 pairs of chromosomes. Then, first we will see the basic features of the human chromosome. Actually, the chromosome can be divided into chromatin and chromosomes during different times of a cell cycle. So chromatin and chromosomes are essentially different forms of the same substance. So they are the same substance in different cell cycles and performing different physiological functions. From picture, we can see the chromosomes found only during cell division because during the cell division, the genetic materials in our cell must be divided into two daughter cells. So the chromosome tightly packed the DNA and ensure that they can be divided into two daughter cells equally. And the chromatin found throughout the interface. And DNA now, during this time, the DNA is being used for macromolecule synthesis. So during the interface, the DNA can be used as a template to synthesize message RNA and then translate into protein. So first, the chromatin. Chromatin is the extended DNA protein fiber in the interface nucleus. And the chromatin, according to their transcriptional activity, the chromatin can be divided into urochromatin and heterochromatin. Urochromatin is low helicity and it can be done with cellular staining. It has some transcriptional activity, but the heterochromatin is high helicity and it is deep staining, almost no transcriptional activity. So the urochromatin has loose chromatin structure and active for transcription, but the heterochromatin has condensed chromatin structure and is inactive for transcription. From the lower picture, the down picture, you can see the heterochromatin and the DNA is packaged tightly with the histone. So the DNA sequence can't be used for a template for transcription. But the urochromatin, due to the histone modification, then its structure changed and the DNA sequence is released from the histone. Then it can be used as a template to transcript. The heterochromatin is the intercellular phase is in a state of condension, and it is generally in inert areas and the replication. Although all the DNA sequence can be replicated, but the replication time is later in the heterochromatin area than other chromatin regions. According to According to the properties of the heterochromatin, it can be classified as structural heterochromatin and functional heterochromatin. The structural heterochromatin is non-transcriptional, mostly located in the centromere, telomere, secondary constriction, the distal two-thirds of the long arm of Y chromosome. So we can see the structural heterochromatin is located in centromere, telomere, secondary constriction. Later we will learn the structure of the chromosome. Then you will better understand where is the centromere, the telomere, and the secondary constriction. Then the functional heterochromatin is cell-specific. It is during the developmental stage, and it is reversible coagulation state. The DNA methylation regulation, it is under the regulation of DNA methylation. The common functional heterochromatin is the sexual chromatin, especially the X chromatin. In this table, is a comparison of the characteristics of urochromatin and heterochromatin. It is from the quantity and regions and the staining reaction or DNA replication as condition to conjectivity and his and his chemical protection that or no both the urochromatin and the heterochromatin is a kind of chromatin, so they are composed of DNA and proteins, so their chemical properties is no difference. Next, we will learn the sex chromatin. Sex chromatin is a special structure shown in the nucleus of the interfish, and the X chromatin is an elliptical body with a size of 1 urometer. Here, this picture shows the X chromatin. It also because during the interfish, it is a chromatin state. So, in our first experiment, we also learn how to observe the bar body, also known as X chromatin. The Y chromatin is a strong fluorescent corpuscle with a diameter of 0.3 unimeter. So the Y chromatin is because the distal part of the long arm is heterochromatin. So this area can be stained to fluorescent. But from the size of the X and Y, we can see the Y chromatin is far smaller than X chromatin. Here, this picture shows the X chromatin by I won experiment from the Chinese students. This dark point indicates the X chromatin. Also, in our first experiment, it named the bar body. So here comes a question. Why do alleles of each gene on the two X chromosomes make no more products for women than for men with only one X hemizygote? So we know the woman has two X chromosomes, but the man only have one X chromosome. And the man also has a Y chromosome, but the Y chromosome is very, very, very small, and is a smallest chromosome in human. So the genes on the Y chromosome is very, very little, and most of the genes encode the main physiological protein. So, but the woman has two X chromosomes. In this condition, the woman should have more products than men. But actually, the woman has no more products than men. Why? We have learned about this point in our single gene disorder affecting factors. Maybe you have forget about it. So it is a long hypothesis. Line hypothesis, also named 莱昂假说 in Chinese. Line hypothesis. Inline hypothesis. There is one X chromosome. It's a inactivation earlier in the embryonic development. And the X chromosome inactivation is random. So maybe from the X from his father or mother also can be inactivated. And once the X chromosome is in activation, the inactivation your gene is completely, so it will have no products, gene products. And once the activation is occurred, the inactivation is permanent and clone reproduction. It is for cell division in an individual. So, because of one of the chromosome of women is inactive earlier in the embryonic development, so his gene products is no more than men with only one X chromosome. And here you should pay attention that in human beings, the X chromosome inactivation is random. But in other species, this inactivation is not random. Next, we will learn another part of the DNA and protein, the chromosome. Oh, here, sorry. It is not chromatin, it is chromosome. The chromosome is composed of countlessly repetitive nucleosome subunits. Here, the nucleosome. It is the smallest unit of a chromosome. The nucleosome is an octamer composed of 4 histones with 2 molecules each, surrounded by 146 base pairs DNA double helix. Here, this picture, the nucleosomes. The histone, the octamer is H2A, H2B, H3, and H4 with 2 molecules each. Then the histone H1 is located on the DNA surface of the junction region of 2 adjacent nucleosomes. Here, the picture shows the H1 histone. So the nucleosome is the smallest unit of the chromosome. Then it condensed package to the solenoid with 6 nucleosomes. Then it forms a chromatin fiber with a 300 nanometer diameter. Then it forms a chromatid with a 700 nanometer diameter. Then it reached the metaphase chromosome during the division cell cycle. So the chromosome during the during the forming process of a chromosome, it has 4 steps. 1 is the nucleosome, 2 is the solenoid, 3 is the chromatin fiber, then the 4 is the chromatid. Next, we will see the number structure and morphology Morphology of human chromosomes. Here we should know two phrases. One is the chromosome set. What is a chromosome set? It is a complete set of chromosomes contained in a normal germ cell. It's called a genome. The chromosome set is in a normal germ cell. So, in human beings, how many chromosome sets in human? Two. Yes. We human beings have two chromosome sets. And in a chromosome set in the human being, how many chromosomes? Do you know? In a normal germ cell, it is a gamete. It is 23 chromosomes in a chromosome set in human being. And the total number of genes in a genome is a genome. Oh, sorry, here is a mistake. Sorry, I made a mistake. Here is the complete set of chromosomes contained in a normal germ cell is called a chromosome set. Then the total number of genes in a chromosome set, named the genome. So, the number of human chromosomes, we have two chromosome sets in human beings, and each chromosome set have 23 chromosomes. Because we have two chromosome sets, so we have 46 chromosomes in human beings. And from this picture, you can see here is XY, maybe the XX chromosome. And here you should know, pay attention. Here, the 21 chromosome is a smallest autochromosome in human beings. It's not the 22, it is the 21 chromosome is the smallest autosome chromosome in human beings. From here, here's a carrier to a diagram of human chromosomes. And from the this picture, we can clearly see how many chromosomes in human beings, and is here a woman or a man? Or did his chromosome have some abnormal? Also can be seen in this picture. So, if we want to know whether the chromosome has some abnormality, then we will learn the structure and morphology of a chromosome. From this picture, we can see in a chromosome. Here, the linkage of two chromatids is a centromere. The centromere, also named the primer constriction. Then, in two ends of the chromosome has the telomere. Here is the 2,6 chromatids, and from the centromere, the chromatids can be divided into short arms, abbreviated as P, and a long arms, Q. Also, in some chromosomes, because the centromere is almost on the end of the chromosome, so it has the short arm of this kind of chromosome, also named as the satellites. And between the satellites and the centromere also have a secondary constriction. And the secondary constriction of a satellite, also named the NOR, the nuclear organization region, the nucleosome organization region. So first we will see the centromere. The centromere, also known as the primary constriction. It is a cellular constriction region on a chromosome where two chromatids copied on one chromosome are linked. Here is the centromere. And the secondary constriction refers to the site of the secondary constriction except the primary constriction. The secondary constriction of the appendage is also called the nuclear tissue region. And this picture shows the general location of the secondary constriction on the human chromosome. So you can see in the 1, 9, and 16 chromosome, it has a secondary constriction near the centromere, and in other chromosomes, it also have some other secondary constriction. Here is a satellite. The chromosome. Here is the D group. The 13, 14, and 15, and the 21 and 22 chromosome have the satellites. So it is 5 chromosomes in human beings have the satellites, then have the secondary constriction named the NOR. The telomere. It is the end of a chromosome that protects the end of a chromosome from degradation and fusing with the ends of other chromosomes. The telomere shortening is associated with the aging of body cells. So, in each replication of DNA, the telomere will have some short. Then, if the chromosome have no telomere, then this chromosome will be degradation. So the telomere is a protector of the chromosome, and if the telomere will not be shortened, then the chromosome will be replicated for many times. And the telomere is a protein DNA complex, and the DNA sequence in the telomere is TTAGGG. or TTT, GGG, TTT, GGG, repeat. Oh, TTA, GGG, repeat. So here you should remember that the telomere is a protein DNA complex and with a DNA sequence with TTA, GGG, repeat. Because the chromosome can be divided into the short arm and the long arm, because of the location of the centromere. So the human chromosomes can be divided into 4 types according to the position of the centromere. One is the metacentromeric chromosome. Then as M is a sub-metacentromeric chromosome. Then the acrocentromeric chromosome. Finally is the telecentromeric chromosome. Finally, telecentromeric chromosome, also named the Y chromosome. And the acrocentromeric chromosome is 13, 14, 15, and 21, 22 chromosomes. If the centromere is the middle of the chromosome, it named the metacentromeric chromosome and the sub-metacentric chromosome. Also, we learned the cells promoting. So we will learn the sex chromosome. The sex chromosome also include the X and Y chromosomes, and on the Y chromosome, it has the pseudo autosomal regions and the Y-specific regions, and the testis determining factor. So it is the Y chromosome that determines the sex of human. And so human is the XY sex determination. Is a XY type. It is a Y chromosome that determines a man or woman. If children have the Y chromosome, then he is a boy. And if he has no Y chromosome, has the X chromosome, then he is girl. She is girl. So the human sex determination is XY type. Also, at class, you can search the internet for some other sex determination type. They have the ZW or ZOXO or some other species. The sex determination of some other species will, according to the temperature or his how to say the temperature or the age of a species. So after class, you can search this, the sex determination type in nature. Learn some fundamental knowledge of the chromosome. Then we will learn the chromosome grouping and heterotype and binding techniques. And this, the karyotype and binding techniques is the most important composition of a chromosome disease. In human beings, the chromosome can be divided into 7 groups named the A, B, C, D, E, F, G. And here, in this picture, we can see A, B, C, D, E, F, G. And the sex chromosome the X chromosome can be included in the C group, and the Y chromosome can be included in the F group, G group. So here you should remember that the human being's chromosomes have 7 groups: A, B, C, D, E, F, G. And the karyotype description is the total number of chromosomes with a dot, then the sex chromosome composition. So the karyotype is an organized picture of the chromosome of a human arranged in pairs by size from largest to smallest. And in human beings, the 1 to 22 pairs called the autosomes, and the last pair are sex chromosomes. And how to describe a karyotype? Here, in human beings, for example, if he is a female, if she is a female, then is the 46.XX. Then if he is a male, then his karyotype is 46XY. So here, you should pay attention, remember the description of a karyotype of a human in this box, in our PPT. This table shows the human karyotype grouping and morphological characteristics of each group of chromosome. The group number is A, B, C, D, E, F, G. Then, here you can see the A group contains the 1, 2, 3 chromosomes. The B group contains the 4 and 5 chromosome, and the C contains the 6 to 12 and X chromosome. Then the D group has the 14, 16, and 15 chromosome. And here you show that this group, the D group, have the satellites because it is the centromere of the D group is ST. And here the E group contains the 16 to 18, and the F group contains the 19 and 20 chromosome, and the location of the centromere is middle. Centric. And the G group contains the 21 and 22 and Y chromosome. It is the smallest chromosome. And the 21 and 22 chromosome had the satellites. Here, this picture shows the unbundled karyotype diagram of human chromosomes. But in this condition, we can also see the size of the chromosome, but we can't see some structure of the chromosome. Then here we should need to know the microstructure of a chromosome. Then we can see some abnormalities or their grouping with their fine structure of the chromosome. So here we should know the chromosome band type. Then we should use the banding technique to get the banded chromosome. In the past, due to the limitation of technology and methods, chromosome research have been affected to a certain extent. It was until the 1956, Levin and Tigel applied Cochrane and hyperosmotic techniques that making the beginning of the modern cytogenetics. So, if we want to get a chromosome band, first we will get the chromosome specimens. The preparation of chromosome specimens requires obtaining a large number of metaphyses because during the metaphase, the chromosome has the best state for us to observation, and the correction can inhibit the synthesis of the Cochrane can inhibit the synthesis of spindle silk protein, then inhibit the activity of the metaphase and stop cell division in the metaphase, so as to obtain a large amount of metaphase. Because low acidity fluid can make the cell volume expand, rupture help to disperse the chromosomes. After friction with a fictive solution or the Carnot solution. Here is the Carnot solution. And this here, the Carnot solution, this procedure we will learn in our experiment, the secondary experiment. Then the tablets were dropped and stained with the giemsa dye to obtain unbanded chromosome species, but with some different process of but if we have our this if the chromosome species. Specimens die with the then with the GIMSA. Then we will get the unbundled chromosome specimens. But before we send the chromosome, we will do some process. Then we will obtain the bundled chromosome. So what is chromosome binding? The chromosome bands are the horizon bands of light and seed or chromosome bands along the long axis that have been processed and dyed with certain dyes, also called chromosome bands. And chromosome binding technology is development on the basis of non-binding technologies. It can display the finer structure of the chromosome itself, which is helpful to actually identify each chromosome and diagnose chromosome abnormal disease. Here, this picture shows the chromosome band of each chromosome. So the binding technology mainly include the gene binding, C binding, Q binding, R binding, T binding, N binding, and high resolution chromosome analysis techniques. Here you should, from this picture, we see the Q binding develops the earliest, and then the G and C binding of a chromosome. The binding techniques. So in human, next we will see the types of human chromosome binding technology. First is a Q binding. The Q binding is dyed with stained with the neutral mustard quinacridone, the QM dye. Finally, it with a fluorescent band. So we should use the fluorescent microscopy to observe the chromosome, and because it is a fluorescent band, so it easily will lost the fluency. So this bending technique has some shortcomings. Then next is the gene band. Gene band do before gene cell staining. It will have some treatment with alkali and trypsin treatment. Then stain with the gene cell dye. So we will see the G band. And the G band is the same with the Q band, but the Q band is a fluorescent band and the G band is the gene cell band. And the G band can be observed under a lighter microscopy, so it is easy for us to observe it and preserve it. And next is preserve it. Next is the R band. The R band with some phosphorylate and high temperature treatment, then stain with the giemsa dye. So the R band is opposite to G band. From this picture, you can see the light area in the G band is the dark band in the R band, and the dark band in the G band is the light band in R band. So the G band and the R band are the opposite bands. Next is the T band. After heat treatment, the giemsa staining shows the telomere at the end. So the T band only dyes the telomere of the chromosome. Here in the middle picture, I see only the end of the chromosome is dyed. And the C band with some alkali treatment. Then with giemsa staining, it dyed the centromere, second constriction. So the C band is with the sodium hydroxide treatment. Or the barium hydroxide treatment. Then we stain with the giemsa dye, and the centromere and secondary constriction is stained. Here is C band. You can see the centromere and some secondary constriction is stained. And the N band, it uses the silver nitrate staining, and the satellite and the NOR areas are blank. So here you should pay attention. The satellite and the NOR, the nucleosome organization region areas, are blank in the A binding technique. So, but we know only 5 chromosomes has the satellite, and then the satellite, the secondary contraction of the satellite, also the NOR. So only 5 chromosomes can be stained into black with the N binding. So here you have 1, 2, 3, 4, 5, 6. So only very little chromosome can be dyed with the N band. And the last is the the last human chromosome binding technology is a high resolution chromosome binding technique. In this technique, the cell is synchronization with the cortisone treatment, and the late prophase and early metaphase chromosome are gated, then with the giemsa dye. Then we will get more fine structures of the chromosome. Finally, we will learn some international system for human chromosome nomenclature. The chromosome here from the high resolution chromosome, you can see there are many regions on the chromosome with some number named. So how to name this region? Here, we will learn. It has some landmark, the region and the band. To describe a particular chromosome band, you need to include the following 4 items. One is the chromosome number. So you should point which chromosome it belongs to. And then the arm symbol, the P or Q, the short arm or the long arm. Then the region number 1, 2, 3, or the band number 1, 2, 3, with no punctuation. So here is the 15q23. So it means the chromosome 15 with the long arm, the short arm. Q is the long arm. The region 2 and the band 3 with no punctuation. And if we want to describe a high resolution banding, we need plus a dot and then write the band number, sub-band number, and sub-band number. For example, the 15q23.12. It means the chromosome, the 15 chromosome, the long arm, and the 2 region 2, 3 band, and the sub-band 1, sub-band 2. So here you should know how to describe the particular chromosome band. And here show the arms are numbered from the centromeres. So here, no matter the P arm or the Q arm, we numbered the region from the centromere. So near the centromere, the region is 1, 2, 3, or 1, 2, 3 from the centromere. Here you should know. Also the high resolution band also named from the centromere, both the short arm and the long arm. Finally, you should see some symbols and terms commonly used in karyotype analysis. So here in our today's class, we learn some knowledge of the chromosome, the chromosome chromatin. But in our medical genetics, you should know the karyotype and how to describe a karyotype. Then, how to get a chromosome band. Then, how to describe a chromosome band. These symbols and terms will use some karyotype analysis. Maybe in our today's class, you should know the A to G group, the 1 to 22, or some number, or the arrow from 2. The P short arm and the Q long arm. Then in next class, in the chromosome disease, you should know this is a break or two is a break and a reconnection of the ins, century, delay, inf, or duck. So you should know some You should remember these symbols because we will use these symbols in the karyotype analysis in the chromosome disease. Then finally, in our today's class, we learn the two types of human chromosome, the chromatin and the chromosome. In the chromatin, we learn the urochromatin, heterochromatin, and the sexual chromatin. In the sex chromatin, we learn the Inline Hypothesis because the human the women gene products is normal than men. And in the chromosome, we learn the structure, composition of the chromosomes, and we also know how a chromosome is composed. From the nucleosome to the chromatids, the nucleosome solenoid, and the chromatin fiber, then the chromatid. Then we should know the sex determination of human is SY type. Then after class, you should search other sex determination type. Last, we learn the category analysis, the grouping, the chromosome binding, and the chromosome binding technology, and the chromosome nomenclature. So today, maybe the karyotype analysis is hard for you. So after class, you should learn more knowledge and have more review for the karyotype analysis because last class we will learn the chromosome disease. Then it's harder than the karyotype analysis. So today's class is over. Do you have any question?
没有问题，老师。No.
Okay, class, I will send the PPT to you.
好的，老师。
跟老师。